Okay, team. Today we are going to do the liquid chromatography lab where we take some plant pigment, which we're going to use a geranium leaf. You can see I've chopped up some leaves in here. And the first part of this process involves using ethyl alcohol to make a crude pigment extract. So what I'm going to do is I'm going to add some of this pure ethyl alcohol. It's 200 proof to the leaves, and I'm going to cook them up on the Bunsen burner. You can see here I have a rolling boil on the ethyl alcohol. Don't want to forget about the ethyl alcohol. Okay, so now I've siphoned off some of this crude pigment extract, which is basically just the ethyl alcohol, and now it has the pigments dissolved in it. And what I'm going to add now is some potassium chloride. You could use sodium chloride, but potassium chloride seems to work a little better. I'm going to add about 1/3 the volume of my liquid, and so I have 6 milliliters of the pigment extract, so I'm going to add 2 milliliters of the KCl. Now, it turns out that plant pigments are very, relatively speaking, non-polar molecules, and we'll talk more about this, but what we need is a good nonpolar solvent. And so now to hyperconcentrate the pigments, what I'm going to do is I'm going to add about 2 milliliters of petroleum ether. Petroleum ether is a very strongly nonpolar solvent. And so what's going to happen then is I added the salt to deal with the water that's in the pigment. The water should go to the salt, and then the pigment should go into the petroleum ether. That's the goal. So I'm going to add that. Now what you can see after I added the petroleum ether and gave it a good shake, you can see how all that pigment, ether floats on the top of the alcohol and the water mixture. And you can see now all of that pigment is kind of hyper concentrated in the very top layer where it's floating. So now what I want to do is I want to siphon that off, and I'll have this really concentrated pigment of pigment extract essentially. Okay. So now I'm going to siphon it off with a pipette. And I'm going to put it in this vial. And there's my very concentrated pigment. So the next part of the experiment involves running the actual liquid chromatography. And what you see here is a piece of filter paper. I'm going to make a little mark right at about 1.5 centimeters on the paper. And that's going to tell me where to apply my pigment. And then I'm going to use a capillary tube, and I'm going to dip it into the pigment solution. Hopefully it's long enough. Yep, and I'm going to apply it onto the line, like so. I have a nice concentrated pigment solution here. So this should actually work out pretty well. The ether allows it to dry very quickly. So I'm going to do one more little row there. And you can see now that's a mixture of the pigments on the filter paper. The last part of the experiment involves taking acetone and petroleum ether and making a chromatography solution out of them. It's going to be a 9-to-1 ether to acetone solution. So 9 parts petroleum ether to 1 part acetone. And then I'll drop my filter paper down in there, and we'll run it in this little vial here and see if we can separate the pigments out. Okay, so now I've mixed up my solution, and it's in the bottom of this little test tube, and I'm going to drop my filter paper down in there, and hopefully we'll get a nice separation of pigments. Let's see what happens. So the solvent is rising up this filter paper. It's just cotton paper. You can see all those pigments rising up, and the idea is they're going to come out of solution based upon their polarity. Because the ether is so polar, the more polar the pigment is, the longer it takes to come out of solution. Okay? So the least polar ones are going to come out of solution first, followed by the more polar ones. And the pigments we're dealing with here are actually chlorophyll A, chlorophyll B, xanthophyll, and carotenoid. And here's the thing, y'all. They're different colors, okay? Chlorophyll A is very blue-green in color. Chlorophyll B is more of an olive drab. Xanthophylls are yellow and carotenoids are orange. And so hopefully we'll get to see these actual colors on the paper. You can see we've already got some pretty good separation at the bottom. You can see the two different colors of green, and once it's finished running, we'll look at it a little bit more. Okay, so it worked like a charm. You can see these different layers, and I'm going to go ahead and pull it out now, and we'll take a closer look at it. And you can see, if you look closely, you have kind of a blue-green color, you have kind of an olive drab color that goes into then a more of a blue-green, and then a yellow, and then an orange. And so if we wanted to label these, it would look like this. We have chlorophyll B, it's the first pigment, followed by chlorophyll A, followed by xanthophyll, followed by carotenoid. So what does that tell you? Well, the least polar must be chlorophyll B and the most polar must be carotenoid. The ether being such a polar solvent, it means that, the more polar the molecule is, the less likely it is to come out of solution. And so the carotenoid just keeps rising up the paper, whereas the chlorophyll B is the least polar molecule. Yes, the least polar, the most non-polar. That's correct. Okay, so that's the quick and dirty on the lab. I hope you enjoyed my little fast video. Have a great day. There's one last thing. I almost forgot. Some of these pigments will phosphoresce under a black light, so they're actually giving off electrons. We've broken down this system of the electron transport chain, and so these electrons are getting excited, but there's nowhere for them to go, and they're literally getting whizzed off into space. And so we'll stick this under a black light real quick and take a look at it, see if we can see the phosphorescence. Okay, so now we are under the black light. I'm going to shut the light out. I don't know how this is going to work out, but we'll see if it works. Oh, it works great. Look at that. So you can see there's the pigment extract, and you can see how it looks. It looks like vampire blood. It's literally phosphorescing. And then if you look at these pigments, you can see which ones phosphoresce more than others. If you look closely at that. You can see the carotenoid really phosphoresces a lot, and the chlorophyll B does as well. Chlorophyll A not so much. Okay, so now we've seen it all. We've seen the phosphorescence as well, and I'm glad I remembered that to put in the video. And you can clearly see these different bands of pigments. That's liquid chromatography to separate plant pigments. I hope you enjoyed it. Bye-bye.